Thank you for the introduction. Today I will be talking about our algorithm multiple approximate kernel learning. We are trying to make it much faster, but it is currently fast and interpretable for bulk RNA sequencing analysis or single-cell RNA sequencing analysis. And this is a joint work with my PhD student Begum, and she did most of the work, but she was not able to attend the conference. Chidam was also a PhD student in my research group. Okay. First, I would like to give you some motivation. First of all, for modeling genotype and phenotype relationships, we need complex machine learning algorithms, especially nonlinear machine learning algorithms. Usually these are either kernel machines or tree-based algorithms such as boosting or bagging algorithms, random forest, XGBoost, or some similar algorithms. And of course, deep neural networks. We need some complex algorithm, nonlinear algorithm to capture complex biology. But these algorithms come with a high computational complexity, of course. So we have to make them more and more efficient if we would like to analyze much bigger data sets. So in computational biology, of course, with the technological improvements, we have now increasing data set sizes. And also, while we are analyzing these data sets, we also extract relevant and meaningful knowledge from data. Okay. Today I'm going to talk about our algorithm, multiple approximate kernel learning algorithm. And we have two main ingredients. One of them is kernel approximation to make it much, much faster. And the other one is also the second step of the algorithm is group lasso to extract meaningful information from the biological data set. So I will explain these two ingredients first. Then I'm going to explain how we integrated these two into a conjoint model. Okay, so we have a genotype measurement data matrix. So we have single-cell RNA sequencing or bulk RNA sequencing data set, which we denote by X. So we have N observations. This might be the patients. This might be the cells. And we have D features, D expression values. So this matrix is our input data. And also we have some phenotype measurements, which is denoted by Y vector. Okay, so we are in a regime that N and D are both large. So most of the algorithms that I mentioned before become very slow. So we have to make them much, much faster by using some computational tricks. First trick that we are going to integrate into our conjoint model is kernel approximation. So in kernel-based methods, we used an implicit mapping from input space to an unknown feature space, denoted by this phi mapping function. At the end of the mapping, we have a kernel matrix, which is a square matrix for the training data and size of n by n. So as we mentioned earlier, n is a very large number. So storing this and also making computations on this matrix is quite heavy. There are approximation methods. There are several approximation methods, of course, including Nystrom approximation, which was mentioned in a talk 2 days ago. And there is this random Fourier approximation. And we use these random Fourier features developed by Rahimi and Recht in 2008. And also it was awarded by NeurIPS conference 10 years later. So it has a very basic idea. Instead of calculating the kernel matrix explicitly, we transform our input data into another space, which we denote by Z, the Z matrix. And this Z matrix will be given to a linear algorithm, such as linear regression or logistic regression. And they empirically and also theoretically showed that if we use enough number of dimensions, we can approximate the original kernel machine. So I'm going to use Gaussian kernel as an illustrative example. So we know that from many earlier studies, Gaussian kernel was shown to be a very good, reasonable choice for modeling genotype and phenotype relationships. So we have these exponent terms, we have the Euclidean distance within this exponent term, and we have the sigma parameter. Now, if we use random Fourier features, basically we don't have any learning, we have a projection step. Instead of calculating the kernel matrix, basically we draw random samples from a normal distribution, and we draw random samples from a uniform distribution as we denote in step number 1 and step number 2. And the Z matrix can be calculated using these trigonometric functions, cosine and sine functions. And the good thing is that if you feed these Z matrix into a linear regression method or a logistic regression method, you are going to get a good predictive accuracy. And this is a very assuring property of these random Fourier features. So I would like to convince you first on a toy data set. Let's assume we have 1,000 data points, and 500 of them are from positive class and 500 of them are from negative class. So this is a binary classification problem, okay? First, I'm going to show you that we can approximate the kernel matrix. So this is the original Gaussian kernel calculated on this data. So I am showing a smaller part of the kernel matrix for visualization, 100 data points. Now, if we use random Fourier features using just 5-dimensional projections, we are going to obtain the kernel matrix on the right-hand side. And as you can see, it's not very bad, right? And if we increase the number of dimensions that we use to 10, it gets even better. And if we increase the number of dimensions to 20, it's getting better and better. So we are able to approximate the kernel matrix, but the thing is that we are not calculating the kernel matrix anymore. We are just calculating the Z matrix, the projection matrix. So we should be able to show that this Z matrix is giving us a good empirical performance, predictive performance. Now this is the support vector machine classification algorithm with the original kernel. I am using 1000 by 1000 Gaussian kernel to find the discriminant between positive class and negative class. And if you use just 5-dimensional projections, and if you use the logistic regression, using these just 5 features, which will be very fast. So the Z matrix is now 1000 by 5 or 1000 by 10. So it will be very fast. So if you look at the purple line, it's a very good approximation of the black line, okay? Now, if we increase the number of random features that we use to 10 or 20, so it's a good approximation of the black line. Of course, we don't expect to get the same result because of the information loss in the projection step, but we are getting a pretty good approximation of the original discriminant. So, of course, this is a toy data set. We have to show these on a real-life data set, and I'm going to mention 3 data sets later. Okay, the second ingredient of our algorithm was group lasso. Group lasso is an extension of the original lasso algorithm. In the original lasso algorithm, basically we have an L1 norm that selects features, that eliminates some of the features, and that picks some of the features during the learning process. Now we need group lasso because we would like to integrate pathways or gene sets as prior biological knowledge into our model. And we will consider them as feature sets in our group lasso formulation. As a reminder, this is the general formulation of the group lasso. We have a loss function between the phenotype measurements and the predicted phenotype measurements using the feature sets. Now I'm going to call them feature sets. And now the difference between group lasso and the original lasso algorithm, now we are not picking features, we are picking sets of features. So some of the sets will be used and some of the sets will be discarded thanks to L1, L2 regularization on the coefficients. So we can solve binary classification problems using logistic laws. We can solve regression problems using square laws in the same formulation. But in this study, we solved 3 different binary classification problems. Okay, how did we integrate these 2 ingredients into a single conjoint model? So we have a 3-step procedure, and I'm going to explain them step by step. In the first step, we have sample by features genotype matrix, the feature matrix. Now, from this feature matrix, with the integration of a pathway database, we extracted subsets of the features. So we are taking slices of these input matrix into P different matrices. Please be careful. Some of these slices might be overlapping, of course, because some of the genes will be present in more than one pathway. In the second step, we used random Fourier features basically to generate these Z1 until ZP. We generated smaller matrices. Maybe in the illustration it is not very visible, but these matrices are much, much smaller compared to the kernel matrices, because kernel matrices are n by n. These matrices are n by 2D, and D is usually a very small number such as 100 or 200. Okay. Now, in the third step and the last step, we concatenate all of these Z matrices into a single input matrix, which will be fed into group lasso. And the group lasso algorithm will make the elimination of the irrelevant pathways, and it will pick some of the pathways as relevant feature sets. So this will summarize the overall algorithm. So if we look at the algorithm in a more structured way, the input is the genomic data matrix X, the feature sets F1, F2, Fp. These might be pathways or gene sets, and the phenotype measurements, which is vector Y. In the first step, we split the genomic data matrix into X, X into P, as I mentioned earlier, possibly overlapping partitions. Then we calculated the low dimension approximation matrices. Then we concatenate these into a single input matrix. Then we solve the group lasso problem. Again, as I mentioned earlier, this can be a binary classification problem or a regression problem. It doesn't matter. Okay, let's come to the real life experiments. We addressed 3 binary classification problems. The first one is early and late stage cancer classification. We have more than 5,000 cancer patients coming from 15 TCGA cohorts, and we have around 20,000 gene expression features. In the second data set, we have 2-year survival classification. We are trying to predict whether the overall survival time is greater than 2 years or not. Again, more than 5,000 patients from 20 TCGA cohorts and around 20,000 gene expression features. The third data set is a single-cell melanoma immunotherapy data set. We are trying to predict whether the cell is a malignant or non-malignant cell. We have more than 7,000 cells with malignancy information and more than 23,000 gene expression features. As I mentioned earlier, these algorithm is quite fast, but the data set that we perform the experiments are medium size, let's say not very large scale, but I will mention the timing of these algorithms later, and our algorithm can also extend to much larger data sets. Okay, we also use 2 different pathway and gene set databases. One of them is Hallmark, which is designed for cancer specifically. There are 50 gene sets, and their sizes are between 32 and 200. And the other one is pathway interaction database. There are almost 200 pathways, and mostly molecular signaling and regulatory pathways. And their sizes are between 10 and 137. Okay, I will briefly mention about the experiments. We performed 100 bootstrap replications with stratification. We have 80% for training and 20% for test. We have inner cross-validation for hyperparameter tuning. And the baseline algorithm is selected as XGBoost. because it is widely used in bioinformatics community for complex problems. And we picked the parameters as shown here, and we picked the maximum depth parameter using cross-validation. For our algorithm, for solving group lasso, we used group lasso R package, and we selected the lambda parameter, which shows us the sparsity of the solution using cross-validation. And the predictive performance measure is used as area under the ROC curve. Okay, first I would like to show you classification results. So on the x-axis, we have the number of selected pathways. Please remember that there are 50 pathways in 50 gene sets in total in the hallmark data set. So by using just 20% of those, we are able to beat XGBoost's predictive accuracy. And if you use more pathways, more than 20% pathways, you can also beat XGBoost predictive accuracy. At the same time, you can identify which pathways are important for the prediction. There are two dashed lines, horizontal dashed lines. One of them is XGBoost using all of the features. The other one is XGBoost with the features included in the pathway database. We have very similar results for 2-year survival classification, even better results. We are able to reach XGBoost accuracy, predictive accuracy using just 10% of the pathways. And if you use a little bit more than that, you can easily beat XGBoost predictive accuracy on this 2-year survival classification problem. There are also other results which are reported in the supplementary material of our paper. You can check them later if you want. Now I would like to also mention feature selection capability of the algorithm. So here I am reporting pathway selection frequencies on the single cell melanoma immunotherapy data set. So there is a pathway called CXCR4, and it is included in the group lasso selection with 100% frequency. And if we check the literature, CXCR4 inhibition is a very well-known pathway in very well-known biomarker in melanoma. So there are also some ongoing clinical trials for this particular pathway. And there are also 2 other pathways selected with almost 90% frequency. So as you can see, in addition to predictive accuracy, we can also extract relevant information from the pathway selection frequencies. Okay, let me summarize very quickly what we have done. We proposed an algorithm called MAKL. It integrates kernel approximation and group lasso. We can include pathways into the model. We did empirical evaluation on 3 binary classification data sets. Compared against XGBoost, we obtain better or comparable predictive performance using fewer expression features. And now I promise you to discuss the computational complexity. In any of these 3 problems, our algorithm is able to return results in less than 30 seconds. Whereas XGBoost takes at least 1 minute, up to 4 minutes, depending on the size of the data set. It's not included in the paper, but we are also able to scale up until 100,000 data points in the order of minutes. Okay, so the take-home message is this algorithm is quite appropriate for large-scale genomic data, such as single-cell sequencing with large sample sizes and with many, many features. Okay, our implementation is available at Begüm's GitHub repository, and also there is an R package available called MAKL. You can easily try the algorithm on your own data sets. Thank you very much.
Thanks, Mehmet. Questions?
Thanks for this great talk. If I understand the equation for the Fourier features correctly, you already need a feature matrix as input for it. Is this right? Like you cannot use it for, let's say, k-mer kernels on sequences or something. You don't have a feature representation of the data.
Yeah, you are right. We need downstream features, not raw sequencing data.
And maybe another question. So usually sparsity is not really improving the model performance, but rather like you need it for interpretability of the model. But in your case, if you have random Fourier features, they are not interpretable anyways. So why do you need the lasso model at all?
Okay, lasso model is for interpretation because lasso model is on the pathways.
Okay, so
if we apply
basically
random Fourier features on each pathway separately, we are going to obtain another feature matrix, but we don't know which one is relevant to the
basically
problem.
in the end, you want to tell the user which pathways are important for the
Yes,
okay.
exactly.
Thanks.
Group lasso is picking which projection is important.
Yeah, thanks.
No problem.